Hi, let's do one update today. Actually, no, we'll do two updates. Yeah. First one's a really cool paper, and it is about, well, it's talking about the furin cleavage site. And this is something that you will see coming up a lot on like conspiracy talk shows and websites and stuff. This is supposedly the evidence that this virus was man-made by evil humans in a lab and not something that occurs in nature. Now, furin cleavage sites occur quite commonly in nature in coronavirus spike proteins and other virus spike proteins. Usually, when they occur, they are associated with higher pathogenicity in influenza. It doesn't necessarily make them more pathogenic in other cases. So there's one called human coronavirus HKU1 from Hong Kong University. Yeah, shout out to them. And that one is really mild. This is a virus that babies get and get over, and don't nobody nobody really realizes that they had it. But it's something that pretty much everybody has had at one point or another in their lives. So it's just a really common but really mild coronavirus. No strong disease, as far as I'm aware, associated with this. But yeah, it's a neat one too. Yeah, a PhD student that's done some work on this. Yeah. Anyway, yeah. So furin cleavage sites. Yeah, they're all over the place. But the genetic engineering angle is that SARS coronavirus 2 has one, and the nearest bat virus, which is different and about 1,000 nucleotides, does not happen to have this. This is a little bit that it looks like has been evolutionarily added in SARS-CoV-2, or it was evolutionarily subtracted in this other virus, which is called RATG13. Which is from Rhinolophus affinis. That's the RA, and I don't know what the TG is. Maybe that's the code for the place where they isolated it. It's from somewhere over in China. Yeah, there was a really nice Nature paper that came out right at the beginning of all of this, and that is still the closest virus, but it's not close. It's just the closest we know about, and it's 1,000 nucleotides away, which is a really long way. Yeah. Okay. So yeah. Anyway, this paper takes a look at the structure of the spike. So in the paper, I should say the title is SARS-CoV-2 and Bat RATG13 Spike Glycoprotein Structures Inform on Virus Evolution and Furin Cleavage Effects. So yeah, fine. Yeah, furin-like enzymes are going to be something that is found inside the lung of people. And so this is an adaptation that would make it more likely to actually work in people. But, yeah, the human-only assumption, yeah, is woefully short-sighted, because there are other animals in this world. There are other kinds of mammal. There are actually a lot of other kinds of mammal that far outnumber humans in this world. Yeah. Bats, for instance. Yeah. And so we've got a virus here that has been in some number of other kinds of animals and has been working its evolutionary way around. And we know that coronaviruses, I think all the coronaviruses we know still come from land mammal. No. There's ocean mammals as well, and there's birds. So there's a couple of birds, but they're a separate group of coronaviruses. And for the most part, it is going to be land mammals that are alpha and beta coronaviruses, which are the ones we're talking about that cause SARS. Yeah. And so there are really lots and lots of other potential hosts out there for which this thing could have adapted. Yeah. And without having to be whatever genetically engineered, all that's it's understandable, but it's also not a high money bet either. Not a good bet, not a good risk, not a good good guess as to the solution. It's a bad hypothesis, basically, because there are so many other ways that it could happen, and there's no evidence that it did happen that way. That's the main thing. So yeah, there are so many other, and we don't know what enzymes pangolins have in their lungs. There are so many different kinds of even rhinolophus bats out there in China. They discover new species of these things pretty regularly. And we don't know anything about, particular enzymes in their lungs. Yeah, we're just only barely getting to grips with like the existence of the species, and all that other stuff, all that other detail comes much later, and only if there's a really good reason. And now I guess there's a pretty good reason, but I think people are still mostly studying the virus in humans and thinking of it in that way rather than thinking big picture that we live in an entire world where, yeah, most mammals have some version of a coronavirus in them, and most birds have some version of a coronavirus that affects them. And these things are just swirling around out there all the time, and occasionally, boom, one of them comes over to people. That's, yeah. I mean, it seems kind of inevitable when you think about it that way, and it seems just weird. All the other stuff that people are throwing in there, but it's stuff you see. This stuff on TV. You've seen it in movies. You've read this bad, bad script how many times or seen it played out. That's what it is. It's this is a human idea that has legs. It's kind of like a meme, basically. And so that's fine. Human ideas. I guess I can't be too hard on those. I'm rather dependent on them myself. But yeah, the furin thing is nothing. Anyway, what's the upshot of the paper? The bad virus looks pretty much exactly like the human version of the virus. 1,000 nucleotides different, but they're still conserving pretty much the same shape. They're showing a couple differences in the way that these things could interact with the receptor. And in general, it's just more information. It's not groundbreaking, but it's neat that somebody's finally working with the nearest bat equivalent to SARS-CoV-2. And that's what I like about it. Thanks very much. This is a Dr. Ben update.